However, such efforts need to strengthen to check the misuses of biotechnology. Biodiversity and its conservations. Biodiversity is a new name for species richness of plants, animals and microorganisms occurring as an interacting system in a given habitat. Biodiversity cannot be replaced. The species becomes adapted in a given habitat after a long course of time. That is why, due to plasticity in their nature and unsustainable resource utilization, over 2.5 lakh species and thousands are threatened to extinction. If a species extinct, it means whole of the gene extinct. The real value of biodiversity lies in the information that are enclosed in the genes, therefore, there is urgent need for future, to protect the genes from destruction. Biodiversity may be defined as the inherent and externally imposed variability within and among the living organisms present in terrestrial, marine and our ecosystems at the specific time. Diversity includes the variability in genes, genotype, species, genera, family and ecosystem at a particular time in a specific region. Thus, biodiversity is an expression of both numbers and differences can be seen as a measure of complexity. Commonly, biodiversity may be considered at the following three different levels. Genetic diversity. Genes are the functional entities of all organisms. They determine the physical and biological features of organisms. Genes of one organism differ from that of the other organisms. Besides, variations arise due to mutation brought about in genome, which is the cause of genetic diversity. Species diversity. A species is a group of organisms which are genetically identical and interbreed to produce progeny. In contrast, horses and zebra are two different species but genetically similar. They can interbreed but produce all infertile offspring. Usually species differ in appearance and thus one can differentiate one species from the other. Thus species diversity is estimated on the basis of total number of species within the discrete geographical boundaries. Ecosystem diversity. There is diverse ecosystems, and organisms living in such ecosystem are adapted to its respective ecosystems. Therefore, there arises diversity among them. Under ecosystem diversity, two phenomenon are frequently referred. The variety of species within different ecosystems, i.e. more diverse ecosystems contain more species, and the variety of ecosystems found within a certain biogeographical boundaries. 1. Biodiversity in India. India is rich in biodiversity to agriculture, animal husbandry, fisheries and forestry. Much has been described in Ayurveda and other ancient literature about the indigenous system of medicine, knowledge and wisdom of people. These are supported by a very strong scientific and technological base. In India, over 5,000 species of plants and animals have already been identified and described. The country is an important center of diversity and origin of over 167 important cultivated plants, spices and domestic animals. A few crops which arose in India and spread throughout the world are rice, sugarcane, bignas, jute, mango, citrus, banana, millets, spices, medicinal aromatics and ornamentals. No country in the world is as rich in biodiversity as ours. Himalaya itself has the natural wealth of plants, many of which are still unknown and many endangered. 2. Conservation of biodiversity. There is an urgent need for biodiversity-rich countries to save it against destruction. However, in most of the developing countries, biodiversity is attached to environment and forest agencies which have no idea it. If such countries are not aware of conserving it for sustainable utilization, they would be to biodiversity and import for well-being of their people. The agreement between Institute of Biodiversity InBio, in Costa Rica and Merck, USA, is hailed throughout the intestinal world. Under this agreement, extracts from the wild plants, insects and microorganisms from Costa Rica are supplied to Merck. In return, InBio receives from Merck over $1.35 million and expects royalty on the commercial products. InBio has to contribute 500/0 of royalty to the government of Costa Rica for National Park Service. The government has given to InBio rights to bio prospects and share conservation work. Thus, the InBip represents an alliance between biologists, biochemists and businessmen. In India, a large number of institutions are involved in conservation and utilization of biodiversity which come under Ministry of Environment and Forest, Agriculture, and Science and Technology. They deal conservation of biosphere reserve, national parks, wildlife, sanctuaries, field gene banks, etc. The country needs more expertise and methodologies besides tiger birdwilled light syndrome. India is predominantly an agricultural country. Therefore, the policy makers have to realize that conservation and sustainable utilization of biodiversity must be placed on the top of all developmental planning. Gene bank, germplasm bank, and plant conservation. Out of 50,000 different plant species, some are already lost and nearly seed plants are threatened to extinction. Due to of natural resource at fast rate, genetic variabilities are being lost forever, which will result in a dangerous future. Therefore, to save the threatened and gradually vanishing species, and to meet the world demand of food, we would have to conserve the natural heritage. The Gene Bank, Germplasm Bank, has taken the challenge of conserving the gene pool of economically, medicinally, and ecologically important plant species. In the first decade of the 20th century, a Russian scientist, N.I. Vavilov, was the first to realize the need of conservation of plant genetic resources. Later, in 1920, he established the first Genetic Resource Center, GRC, of the world at Leningrad. Thereafter, a global concern was raised after the International Biological Program in 1964. 1. World Genetic Resource Centers. During the last 2 decades, many regional and international GRCs have been set up in different countries. International Rice Research Institute, Manila, Philippines, has Rice Germplasm Bank where 25,000 varieties of rice germplasm has been collected throughout the world. Similarly, maize germplasm have been stored at Maize and Wheat Improvement Center, Mexico, with more than 12,000 varieties. Instituto Colombiano Agropecuario, Colombia, more than 2,000 varieties. Instituto Nacional de Investigaciones Agrícolas, Mexico, with more than 7,000 varieties, and potato germplasm at International Potato Center, Lima, Peru. A seed bank has been set up at the National Bureau of Plant Genetic Resources (NBPGR), New Delhi, which is associated with a world network of gene resource centers coordinated by the International Bureau of Plant Genetic Resources of FAO. 2. Conservation and storage. For the conservation of genetic materials, seeds, pollen grains, vegetatively propagating parts, plant tissues or genome of plant cells are collected. These depend on ease to handle, nature of crops, longevity, expertise, methodologies available, space required, problems of genetic erosion of stocks, and expenses. The seeds are compact and easy handle, hence seed collection and preservation are commonly adopted. Seeds are stored in sealed hermetic containers, dried to a desired moisture level and maintained at desired low temperature. Viability of pollen grains differs in different species, the species having maximum viability selected. The families showing the highest longevity of pollen grains are Pinaceae, Primulaceae, Rosaceae and Saxifragaceae. The longest period of pollen storage recorded is 9 years at 200 for apples. The vegetatively propagating parts, corn, bulb, tubers, etc., of some plants such Dioscorea, potato, sweet potato, etc., are used for storage purposes. Techniques have been developed to culture the plant cell, tissue and organs. In vitro grow cultures, e.g. plantlet, apical meristem culture, etc., are stored, see Chapter 10. Storage of in vitro grown cultures has many advantages over the others such as requirement of less space, cheap in maintenance, high propagation potential, least problem of genetic erosion of stock, and maintenance of pathogen-free stock. End of the Chapter 1. To solve the problem statements, see the figure.